Today, I will be talking about characterizing fMRI signatures of systemic physiology in healthy aging. To start, I have no financial interests or relationships to disclose. A large number of fMRI studies have shown that slow variations in systemic physiology can drive significant changes in fMRI signals. For instance, in this seminal study, the authors showed that the respiration volume per unit time, RVT, which is the blue envelope of the respiratory traces, can account for considerable fMRI variance in widespread gray matter regions. In a separate study, the authors correlated heart rate changes with fMRI signals, and we can see significant correlations at different temporal lags. In addition to the respiratory variation and heart rate, changes in end-tidal CO2, amplitudes of pulse and blood pressure can also drive widespread fMRI dynamics. While these physiological signals are commonly viewed as nuisance factors and removed from fMRI signals during the data preprocessing, they also contain very rich information about the blood flow regulation and vascular anatomy. Several recent studies have shown that these fMRI signatures of systemic physiology are not homogeneous across the brain, likely arising from distinct arterial transit time and heterogeneous vessel geometries and densities in different brain areas. In our recent study, we also show that these physiological dynamics can form different clusters that mimic neuronal networks. Timing and shapes of fMRI signals coupled to changes in systemic physiology are distinct across different clusters. Therefore, exploring the spatial temporal structures of the rich physiological dynamics can give us new insights into the brain vascular physiology. The goal of this study is to characterize how these fMRI signatures of systemic physiology altered in aging. Here is some evidence demonstrating age-dependent changes in vascular physiology. Studies have shown that there is a decreased blood flow and cerebral vascular reactivity, an increased arterial transit time and vessel stiffness, etc., with advancing age. In this study, we will test whether some of these age-dependent vascular changes can also be identified in resting state physiological fMRI signals. This may be a very compelling case showcasing that fMRI signatures of systemic physiology holds the promise as a biomarker for brain physiology in more broad clinical investigations. To achieve this goal, we leveraged the big data of the HCP Aging Project. We used the first two resting state scans. Like our previous study, we focus on two types of physiological dynamics, including changes in respiratory variation and heart rate. RV was computed as the standard deviation of respiration amplitudes for each 6-second sliding windows. An inter-heartbeat interval was computed as the average heart rate interval across 6 seconds long windows centered at each TR. 422 subjects with good physio signal quality were carefully chosen for the analysis, and we used age 65 as the threshold to further divide the subjects into two groups, the younger aging 1 group and the elder aging 2 group. To understand the temporal relationship between RV and the global cortical fMRI during resting conditions, we conducted the cross-correlation analysis, and all fMRI signals and physiological metrics were upsampled to a temporal resolution of 200 milliseconds before cross-correlation. The green curve here shows the results estimated from 190 subjects from the HCP Young Healthy Subject cohort, and the cross-correlation between RV and fMRI signals exhibited a biphasic pattern that was consistently reported in the previous fMRI literatures. Global cortical fMRI signal lags RV by around 10 seconds. As the subject age increases, we can clearly see an increase in the temporal lag, and the elder aging 2 group exhibiting a further delay compared to the younger aging 1 group. The scatter plot clearly depicts how the temporal lag increases with age. We then inspected how the temporal lag varies across the cortical regions. The relative intracortical temporal lags are roughly consistent between two age groups. For instance, the sensory regions tend to have faster responses. At the group level, the frontal and parietal areas exhibit the most significant delays in aging. Age-dependent delays are not specific to the cortical gray matter. Similar patterns can also be observed in the subcortex, brainstem, cerebellum, and cortical white matter. We also notice a much slower response in the cortical white matter, and the white matter roughly lags the gray matter by about 5 to 6 seconds. Overall, the lagged respiratory responses are consistent with recent reports of both increased arterial transit time and delayed local vasodilation in aging. The gray versus white matter timing differences are also consistent with delayed vascular transit time in white matter as proposed in a recent study. As showing the spatial patterns of delayed arterial transit time, age effects are also strongest in the frontal and parietal regions. In addition, as you might already notice, our results also show a modest reduction of RV fMRI cross-correlation in aging. However, the difference is not significant between the aging 1 and aging 2 group. Reduced cerebral vascular reactivity is a hallmark of aging. One likely reason that we fail to observe a significant change here is that RV is not an absolute quantification measure. Its amplitude can be affected by how you place the respiratory belts and the SNR of the recordings. We next examine age-dependent changes in fMRI dynamics coupled to heart rate changes. The cross-correlations between the global fMRI signal and HBI also exhibited a prominent age dependence, as you can see. Compared to the HCP young and aging 1 group, aging 2 group exhibited an earlier trough in the fMRI-HBI cross-correlations. It is worth pointing out that the temporal lag for HBI is much faster compared to RV-coupled dynamics. Additionally, a stronger correlation between fMRI signals and HBI was also identified in the elder group. However, when we inspect intracortical distributions of temporal lags and cross-correlation values, they exhibit very distinct spatial signatures. Unlike widespread increases in the coupling between heart rate changes and fMRI, regions exhibiting significant between-group temporal lags are more focal, primarily in ACC and the posterior portion of the brain. Age-dependent changes in fMRI dynamics coupled to HBI is also identified in other brain regions. However, in this case, white matter only lagged gray matter by about 2 seconds. The temporal difference is much shorter than the respiratory responses. So why are cardiac responses faster in aging? One possible mechanism we are thinking is increased vessel stiffness in aging. While the exact mechanisms of how heart rate changes affect fMRI signals are unclear, it is likely that this is through changes in cardiac outflows. When increased blood flow are pumped into the brain, a key factor determining the speed will be the arterial elasticity. Many studies have already shown that the aging population have increased pulse wave velocity and enhanced pulse pressure due to decreased arterial elasticity. Similarly, we may observe faster vascular responses and stronger correlations with heart rate changes. Furthermore, while both response timing and magnitudes are affected by vessel elasticity, timing also reflects other factors such as the brain circuitry involved in autonomic control, and response magnitudes may vary further according to the local vessel density. This explains the divergence in their spatial distribution. We also consider potential alternative mechanisms, ones that decreased fractional gray matter contributions due to cortical thinning, as lots of studies indicated that cortical thickness generally decreases with age. Another is that aging cohorts are more likely to become drowsy or falling asleep during the scan, which could potentially account for the changes in the cross-correlation. There is also possibility that RV-HBI coupling becomes much stronger in aging population, and the respiratory effect will also changes the cardiac responses as well. However, these alternative mechanisms are unlikely the dominating factors for observed physiological responses in our data. We first examined whether cortical thinning could account for increased correlations with heart rate changes. The first row here plots the dependence of cortical thickness on aging. The most prominent reduction of cortical thickness occurs in the frontal and temporal lobes, which are not aligned with regions demonstrating the most prominent increase in HBI correlations. Therefore, cortical thinning is unlikely the major factor underlying the enhanced HBI-fMRI coupling. We next examined the influence of vigilance changes. Several recent studies have shown that when subjects become drowsy, we will see stronger coupling between systemic physiology and fMRI signals, as shown in the plot here. However, this may not be the major reason underlying age-dependent changes seen in our data, owing to several reasons. First, we observed a decrease instead of an increase in RV cross correlations. Second, the arousal effects are found in regions slightly overlapping with those with largest age-dependent effects, such as the visual cortex, but they do not share a completely identical spatial pattern. Third, drowsiness is always accompanied by a drop in the heart rate. However, we don't see a significant difference in the mean heart rates between two age groups. Collectively, arousal effects cannot fully account for the age-dependent changes in the physiological responses. Finally, when comparing the coupling of respiratory and cardiac dynamic between the two age groups, we do observe a coupling between RV and heart rate changes at time zero, which may explain the positive peak of RV-fMRI coupling. However, the correlations are not significantly different between two groups. In summary, our study has shown that fMRI signatures of systemic physiology exhibit prominent age dependence. We observe a like response and a minor reduction in total fMRI variance contribution in RV respiratory dynamic, consistent with increased arterial transit time and decreased cerebral vascular reactivity reported in previous studies. We also observe a faster responses and an increased explained variance in HBI cardiac dynamics, which likely mirrors reduced vessel elasticity in aging. Given that these physiological fMRI signals arise from distinct mechanisms, we are able to isolate and identify distinct spatial signatures of age effects on RV and HBI dynamics. Our results suggest that these fMRI signals can provide rich information about vascular physiology and hold strong promise as a clinical marker that is complementary to widely exploited neuronal dynamics. Finally, our results also hold implications for broad resting state connectivity studies. If we view these physiodynamics as nuisance factors, we will need to account for changes in their shape across the lifespan. Also, since physiodynamics also exhibit age-dependent changes, we will naturally expect that regressing them out of the data will exert on significant changes in characterized brain network patterns. This could be factored into the data interpretation of your results.